What makes you, you? Part of the answer is genetics, the DNA you inherit from your parents. But there's another layer called epigenetics that helps decide which parts of your DNA are used, when, and how much. If genetics is the musical score, epigenetics is the mixing board. The notes don't change, but the volume, timing, and feel can. A gene is a stretch of DNA that contains instructions, usually for making a protein that does work in your cells. Humans carry thousands of genes arranged on 46 chromosomes in most cells. The specific versions of genes you carry make up your genotype. How those genes show up as traits, eye color, enzyme levels, disease risk, is your phenotype. Genes operate through a flow of information. DNA is copied into RNA, and RNA helps build proteins. Variations in DNA, like single letter changes or small insertions and deletions, can influence the shape of a protein or how much of it gets made, nudging traits in subtle or dramatic ways. Epigenetics focuses on how genes are turned on or off without altering the underlying DNA sequence. These control systems help cells remember who they are. A liver cell keeps liver genes active and neuron genes quiet, and it passes much of that pattern to its daughter cells during division. Three broad mechanisms do most of the work: DNA methylation, histone modifications, and non-coding RNAs. DNA methylation places tiny chemical tags, methyl groups on specific DNA bases, often at CPG sites. When clusters of these tags accumulate near a gene's promoter, and the gene typically becomes less active. In many contexts, methylation patterns are stable and act like a cellular memory, helping maintain long-term gene activity states. Histone modifications change how tightly DNA is packaged. DNA wraps around histone proteins to form chromatin, chemical marks added to the flexible tails of histones, such as acetyl or methyl groups, alter how accessible the DNA is. Looser chromatin tends to invite the molecular machinery that reads genes. Tighter chromatin tends to keep it out. Different marks at different places can either activate or repress genes, giving cells a rich code to fine-tune expression. Non-coding RNAs, molecules that aren't translated into proteins, add another layer of control. Some, like microRNAs, dial down the production of specific proteins after a gene is transcribed. Others, like long non-coding RNAs, help recruit or block protein complexes at chosen spots in the genome, steering which genes are used. Why does epigenetics matter? First, development. Early embryos undergo sweeping reprogramming that erases and rewrites many epigenetic marks, restoring flexibility so cells can become any tissue. An exception is genomic imprinting. A small set of genes carries parent of origin marks that influence how strongly they're expressed. As development proceeds, cells lock in new epigenetic patterns that stabilize their identities. Second, the environment can leave biological fingerprints. Identical twins start life with nearly the same DNA and very similar epigenetic patterns, but their marks diverge with age and different experiences. Historical natural experiments add evidence. People conceived during severe famine have shown altered methylation at certain growth-related genes decades later. These findings connect early life conditions with long-term biology, though the precise cause and effect pathways are complex and still being mapped. Third, health and medicine. When epigenetic control goes awry, protective genes can be silenced or harmful ones inappropriately activated. This is common in cancer and appears in other conditions as well. Some approved therapies already target epigenetic enzymes. For example, drugs that inhibit DNA methyltransferases or histone deacetylases to restore more normal patterns of gene activity in certain blood cancers. These treatments don't fix DNA mutations, but they can reshape the gene expression landscape in useful ways. It's also important to clear up myths. Epigenetics isn't mystical, and it doesn't mean that a thought instantly rewrites your DNA. Most epigenetic marks are cell-specific, and many are reset in eggs, sperm, or early embryos. While limited cases of marks persisting across generations are being studied in animals, strong evidence for widespread transgenerational inheritance in humans is limited and remains an active research area. Looking ahead, researchers are developing tools to edit the epigenome with precision. Systems that guide DNA-binding proteins, like a dead version of CRISPR-Cas9, can be fused to activators or repressors and parked at chosen genes, nudging them up or down without cutting the DNA. It's a powerful way to test how gene regulation shapes cells and diseases. Clinical applications are still in their early days, but the concept is promising. In short, genetics writes the score, epigenetics runs the mixing board, choosing which tracks play, how loudly, and when. Understanding both helps explain development, individual differences, and new paths for treating disease. Get your DNA tested along with a strategy for wellness by going to poshfitness.com/DNA coach. Until next time, stay curious and stay healthy. Peter Marino, signing out.